The interest in my lab is to understand how membrane transport proteins work. So membrane transport proteins are integral membrane proteins that span the lipid bilayer across the membrane, and they allow the transport of ions, solutes, metabolites, and all sort of macromolecules. So through the years, we've been looking at different families of membrane transporters that actually underlie physiological processes that are related to very important agronomical traits. And in that process, we've identified different families that mediate the transport of all sort of nutrients and molecules that are associated with this particular agronomic traits. So in that process, we find transporters associated with abiotic, with biotic tolerance to different stresses, plant path responses, mineral nutrition responses, and qualities in terms of fruit quality and the quality of grain and cereals. So the current questions that we're dealing in the lab right now is, when you have a particular family of transporters, say the mate family of transporters, you have a very conserved structure for that particular family. However, the functionality of that family is very broad. I mean, these transporters are able to select or discriminate among a whole range of different substrates. So what we're trying to understand is, how is that function-structure relation of the protein, and how does it work in terms of selecting or identifying these different substrates, which ultimately define the function of this protein. For that purpose, what we do is we take our gene or transport of interest, we express it in a variety of heterologous systems, and we mostly use electrophysiological approaches to study and characterize the biophysical characteristics of these transporters in terms of what do they permeate, what do they allow to move across the membrane, how can they select, how can they discriminate among those different substrates. So electrophysiological approaches, and that's what the remaining of this presentation is going to be. It's sort of the different methodologies we use in order to characterize these transporters. What we do is we measure changes in voltages, we measure changes in currents at a variety of different scales. We can look at what happens at the whole organism, at the whole organ. We can look at what happens at the single level in terms of that transporter, or we can look at the single molecule level as well. So in terms of a whole organ, we can measure surface potentials, changes in the voltage across a large area in a particular organ, or we can measure changes in membrane potential in a specific cell using very fine glass microelectrodes. And these changes in voltages are in response to the opening and closing of these transporters, in response to an environmental cue. Now at the cellular level, we're able to isolate these individual cells and now look at the activity of the transport itself using something called patch clamp technique. And what this allows us is to monitor the transport of these ions across the membrane under different conditions. Now we can also use this approach to actually pull the pipette out, and what you obtain is a little patch of membrane at the tip, and you're able to characterize the transport activity of one or two molecules that are contained in this patch, which ultimately underlie those characteristics that you should be observing at the whole cell level. So this is our sort of single molecule analysis of the functionality of a transporter. We also use an approach called 2-electrovoltage clamping. For this purpose, we express our transport of interest in Xenopus oocytes, which are big cells. They are about 1 millimeter in diameter. We can come with a pair of electrodes and monitor the transport properties of the transporters expressed in the plasma membrane of the cells. What this provides a big advantage is in the sense that we can easily go back to our DNA, cause a mutation, and look what the impact of that structural change is in the functionality of the transporter. And we can do this in a matter of weeks. So it's a very single cell, high throughput way of analyzing what the function and the structure relationship is. With this set of techniques, what we can also do is we can take advantage of using a combination of chimeras with fluorochromes, YFPs, GFPs, to understand a little bit more of the assembly and the stoichiometry of those transporters. For example, we can do a traditional BIFC complementation analysis of our transporters to see how the different subunits of the transporter assemble, or they don't, to form the functional protein. We can also use turf microscopy down at the single molecule level. And with this purpose, what we're doing is, what you can see in this small movie, is the photo bleaching of single GFP molecules upon hitting them with a laser. And we can find a correlation with how many discrete or photobleaching steps we see at the membrane in each one of these spots, and the number of subunits that are being associated. In this example that I'm showing you here, what you see is that there are 2 discrete steps, which mean that the GFPs, you always see 2 GFPs together, which means that this particular protein assembles a dimer, functionally assembles a dimer. Finally, we also work with recombinant protein, so we can get recombinant protein of our transport of interest. We purify it, and we do a functional reconstitution of these transporters back into their lipid environment. Once we have the functional reconstitution, what we can come back is with our electrophysiological techniques and measure the transport that occurs across one of the single molecules. Once we understand and once we know that this recombinant protein actually folds correctly and is able to reconstitute the functionality, obviously we can use that protein to start doing structural analysis such as X-rays and cryo-EM determinations of our transporters of interest. So with that, I just want to leave you with what I think the future is, and it's I think we're at a point where we can start engineering membrane transporters. Doing how, if we're able to understand this relationship between structure and function, we should be able to modulate or change the structure in such a way that we can modulate their function, and ultimately what we change is a phenotypic output mediated by this transporter. So that's a flavor of what we do across the lab, and if you have any questions, I'm happy to answer them.